Our next speaker joins us from the Jackson Laboratory in Bar Harbor, where she's the scientific director. Nadia Rosenthal also holds a chair in Cardiovascular Science at the Imperial College London. She was awarded a PhD from Harvard Medical School, where she later directed a biomedical research laboratory, then established and headed the European Molecular Biology Laboratory campus in Rome. She was founding director of the Australian Regenerative Medicine Institute at Monash University in Melbourne and founded EMBL Australia as its scientific head. Her research has led to significant advances in the development of novel gene and cell-based therapies for muscle aging and heart disease. Please join me in welcoming Dr. Nadia Rosenthal.
This is my favorite new organism, which is a bit of a paradox given that I'm working at the Jackson Laboratory, which most of you know is sort of mouse central. But I'm going to tell you how I believe looking at a number of different organisms and variation in biology can teach you a hell of a lot about genetics. So as we know, I'm a regeneration biologist. I finally found something to do with my molecular biology, and I work on regeneration both from the point of view of being a phylogenetic variable, that is to say, different organisms regenerate to different capacity. In this case, as you can see, the heart, which is my favorite organ at the moment. I think I just screwed it up again. I was just looking for something to point with, but that didn't work. I'm sorry, I goofed. Hearts in fish regenerate extremely well, whereas hearts in, what did I do? Oh, okay, fine. I'll just use this. Whereas hearts in other organisms, such as the mouse, are very poor regenerators, and it's not clear why. It doesn't make that much sense. We need to have a heart, and the fact that it doesn't regenerate doesn't seem to have any obvious advantage evolutionarily. And yet, even within something like a fish, where you think of regeneration as something very sort of fundamental to their biology, we see that even between closely related teleost species, such as the medaka on the right and the zebrafish on the left, there's essentially a complete absence of regeneration in the medaka, so that even within a species, you really see variation in an incredible amount. Now, we also know that during development, regeneration is variable. So at different times during your ontogeny, you are able to regrow parts of your body before you are born. And indeed, even mice at birth are able to regenerate their hearts if you infarct the heart of a newborn mouse. Here you see on the left, 21 days later, a cross-section of the heart looks essentially like a normal heart, whereas if you do that same procedure only 1 week later in the mouse's life, it doesn't regenerate, and it never does. And so there's this obvious regeneration-fibrosis dichotomy that rapidly sets in. And we know also that between individuals, regeneration is variable. So, for instance, heart attack outcome. We all know, as clinicians know, that different people respond totally differently to a heart attack. And this could be because of stress and gender and lifestyle and various goodies, or it could be something genetic in there. So the elephant in the room is the immune system for me, because obviously the immune system controls fibrosis, and fibrosis and inflammation are the two bugbears in regeneration. And so we are interested in the fact that there are key parts of the immune system that seem to be evolutionarily conserved and may vary enough to have some profound effect on our capacity to regenerate. So we've been focusing most recently on the immune system as a way of looking into this problem. And in the case of regenerative tissue, such as here you're seeing the heart, damage to the heart tissue inevitably induces inflammation. And the question is, how do you keep the balance between inflammation, which is an absolute requirement during the early stages of wound response, keep that under control so that cell replacement can persist? So here's the axolotl, who you've seen before. It's a very interesting aquatic salamander that lives, or used to live, I think it's extinct in its natural habitat now, in the lakes of Mexico, in the mountain lakes of Mexico. And this one's an albino, but they usually look really scary and black. And in this case, you can see all the various bizarre parts of this animal, and I can tell you that just about everything other than its entire head can be cut off and regenerate. And this is bizarre because we don't really know why the animal would develop such an incredibly robust response so that you can grow a new limb every time. You can cut it off sadistically over and over, and it grows back over and over. And this is an age-independent attribute. And one of the reasons for this, we believe evolutionarily, is that they are really awful. They eat each other for breakfast, and whether they're crowded or stressed or hungry, sort of like academics. And so we started to look at the immune system of, not academics, of axolotls. And we noticed, James Godwin took this on. It was heroic because we had no antibodies, and so we had to sort of borrow from chickens and fish and people who had worked on axolotls and dead scientists' freezers who had antibodies to axolotls. And eventually we got to the point where we could say fairly confidently there was nothing wrong with an axolotl's immune system whatsoever, or at least nothing different from an organism like ourselves. So the only thing we did notice is that after we amputated a limb, the stump attracted these very same cells, but instead of this progression, this stately progression of pro-inflammatory and then anti-inflammatory cells, it all happened at once, as if everybody came to the cocktail party exactly at the same time. And without that, it appears not to work. So the data that we have so far to prove this is fairly crude because we can't do genetics in axolotls yet. And so what we were able to do was remove the macrophages, which of course are a key component of the immune response to injury, and we did so using a trick called claudinate liposomes. And what we found was, unlike what we saw in the normal situation, where huge numbers of chemoattractants brought the macrophages almost immediately to the site of injury, in the axolotls that had been treated with these claudinate liposomes, depleting them at least for about 3 or 4 days of their macrophage component, you can see on the right that stump. That is what an axolotl's non-growing limb looks like, and that axolotl only lost its macrophages for a few days, but that was enough to completely block the limb from ever growing back because this huge fibrotic scar formed instead. Now, when we injured that scar, amazingly, we were able to regrow the limb entirely. So what that suggests is that as long as you have an intact immune system, at least in the context of an axolotl, it's no problem. I mean, all the mechanical implications of this are not to be discounted, but geneticists don't care about that, at least the ones I talk to. Now, the same thing is true of the axolotl heart. You can injure the heart, it grows right back. Here we see an injured heart on the left, which is sort of cryo-injured with a cold probe, and it grows back within about 2 months. And on the right, you're seeing, I don't know what's happened, my slides are all purple, but whatever. You're seeing a heart that has actually experienced this and has grown back. The cardiomyocytes are replicating, which they don't do in our own hearts, and the animal is full of macrophages, which is, of course, absolutely critical. And what we found in doing the same kind of experiment is the heart's no exception. Without macrophages at that moment when the animal is injured, the scar that forms is acellular and permanent, and it completely blocks heart function as it does in us. So we wondered what was going on, what were the macrophages doing? And one thing we know that they are doing is secreting factors. And indeed, we saw plenty of cardiomyocyte proliferation, which is normally thought of as the big block in our own incapacity to regrow our hearts. But the axolotl can't grow a heart even when it has cardiomyocyte proliferation. So we looked at the culprit, which is macrophages. And what we found was that every function of that particular cell type depends on a growth factor called IGF-1, which happens to be my favorite growth factor. I've been working on it for 25 years. So we sort of focused in on that, and it turns out that IGF-1 is one of many growth factors that macrophages use to modulate their environment. Now, in the case of the normal way you think about this factor, it's secreted by the liver, it controls our growth during early life, and it also responds to injury by enhancing regeneration, but it wasn't clear how. It's FDA approved. So we started playing around with it at first by just making animals that overexpressed IGF-1 and found that we could grow these super transgenic Schwarzenegger mice by overexpressing IGF-1 in the muscle. This got me famous for 15 minutes, and I got a lot of weightlifters calling me. And then that all died down, and I started working on the real science, which was what is it doing? And what it's doing is it's countering inflammation. So to make a very long story short, we've shown that macrophages during regeneration, in this case of skeletal muscle, secrete IGF-1 in a very specific moment, namely at day 5 after the injury, right at the time when the macrophages are transiting from a pro-inflammatory M1 phase to an anti-inflammatory M2 phase. So we asked the question, is this true, and unrelated? And the way we did this was Joe Tonkin in the lab knocked out the IGF-1 gene in a monocyte-specific fashion in a mouse. Now, the mice didn't seem to mind that until you injured them. And once you injure them without IGF-1 being expressed by the macrophages, you got what that little arrow is pointing to, which is a fibrotic mess. And that was because the M2 macrophages couldn't form. And so IGF-1 is this factor that is secreted in a time-dependent way by macrophages and shifts them into this anti-inflammatory stage. And this is true also in the heart, and we've shown this. I'm trying to keep this very, very short because it's all published, to point out that if you have a heart overexpressing IGF-1 and you injure it, the heart doesn't grow normally above its normal size, but it responds to injury in a totally different way. And that's probably because you've reduced the amount of scarring and you've increased the functional capacity, and you've done so by pushing those pro-inflammatory programs right over into an anti-inflammatory and pro-regenerative state. So we can do something about these sort of blockades that we see in our own bodies with growth factors if we know how to approach this. But we were more interested in the mechanism of how the macrophages were actually doing this and what was IGF-1 doing. And we started focusing on fibroblasts for obvious reasons, because fibroblasts respond to mechanical stress. They're also the part of the interstitium that sort of holds it all together. They're understudied, as far as I can tell, other than their capacity to make very nasty scars and fibrosis. So we started to look at fibroblasts specifically in the heart. And the reason we did this was because, of course, the immune system controls how fibroblasts behave. And that's something that's in the literature. Activated fibroblasts are necessary to keep a wound intact, but they also, in the wrong setting, can form a fibrotic scar. So Alex Pinto in the lab started doing unsupervised analysis of cardiac cell composition using an algorithm called SPADE that Gary Nolan developed at Stanford. And this allows you to cluster cells according to what they're expressing on their surface using flow cytometry, and then an algorithm to pull these cells together in groups. And what we found was that endothelial cells were by far the most abundant non-cardiomyocyte cell in the heart, but that fibroblasts were followed along, about 20%. And we were able to sort of look at these fibroblasts in some detail in terms of what they were expressing. And in passing, we noticed that there's been a lot of hoopla about cardiac stem cells and mesenchymal stem cells in the heart being very important for heart regeneration. Well, we couldn't find anything different about our fibroblasts from any report on cardiac stem cells. So we actually think that we are working with fibroblasts that are somehow being attributed all these extra sort of godlike qualities. So I think we should exalt the fibroblast instead of the stem cell, and take on the fact that fibroblasts, especially in organ context, are extraordinarily important. So cardiac fibroblasts also express a huge number of unexpected genes. I've got the pink arrow, oh, they're purple, wow, that's amazing. I've got the purple arrows, or the fuchsia arrows, pointing to a number of genes which, if you have any knowledge of heart development, will stand out as very important genes for cardiac development. And yet these are adult fibroblasts isolated from adult mouse hearts expressing these factors at a very high level. And when we did a cytoscape of all the fibroblasts from a number of different organs, asking the question, is this just true of the heart, or do other organs also have a unique feature of their of their organ-specific fibroblast profile, we discovered that this is not specific to the heart, that skin has fibroblasts that express skin-specific, developmentally regulated genes, liver the same, lung the same, kidney the same. And maybe this, just to connect to Don's talk, is one of the keys for how you end up with organs on a chip where you don't end up with a gut cell when you've started with lung, because you're starting with a wise cell, a cell that's had some information about where it started from. Now, in the middle of this cytoscape are all the canonical genes that are, canonical genes that are associated with things that all cells have to do. But look at how few those are in comparison to the organ-specific genes. And even more bizarre was the fact that in fibroblasts, we seem to be retaining the famous positional Hox code to some extent. And this was partially noticed before, but never quite as kind of overtly as this. And what it looks like to us is that something about fibroblasts retain some of this embryonic information for reasons that probably have to do with their role in regeneration. So my hypothesis at this point, because this is all unpublished work in progress that has not yet been rejected, is that organ fibroblasts respond to immune induction. And they do so by making extracellular matrix. They're probably also responding very directly to mechanical stress. And they secrete signals and recapitulate positional patterns in response to stress and or bioelectric signals, or who knows what else. But there's something in those interstitial cells that is retaining an information of morphogenetic code, and we're trying to figure out what that is. Okay, that's a bit of a digression from the main sort of point I wanted to make, which is that individuals within a species have extraordinary variation in their response. Now, the question, and I'll end with this, the question with whether mouse strains respond differently to MI gets back to a central point that came up in Don's talk, which is that animals often are poor substitutes for humans in terms of recapitulating responses to drugs, responses to infections, etc. And what I would like to argue is that this is because almost everyone in this room is ordering C57 Black 6 from the Jackson Laboratory. Thank you, thank you, thank you. However, I would like to point out we sell other flavors of mice. We are the Baskin-Robbins of the mouse world. And you can buy all these different mice from us. And let me tell you why you should consider this. Actually, probably not. You should probably just collaborate with me and then I can pay for the mice. But you didn't hear that. Okay, homozygous across their entire genome, all mice within a strain are genetically identical, reproduce models of disease. We have spent the last 25 years making sure that all our mice look like this. And yet, if we look at different mice even within our inbred lines, we can see huge differences. So AJ is known to have spontaneous lung carcinomas, C3H has hepatomas, and the poor old C57 Black 6, which is considered the mouse, is one of the sickest animals on the planet. It gets, diabetes, it's neurotic, it gets blind. Okay, all that said, all these things exist in humans, and indeed, much of the human genome in its sort of quintessential format is recapitulated in the mouse genome. Here's just a map of, you can't see because the colors have been distorted, but it matches. However, the classical inbred strains are limited in their genetic diversity. They cannot possibly recapitulate what we're seeing in humans. These are the three most normal strains in Domesticus, Musculus, and Castaneus are the most predominant mouse strains that hang around with humans like dogs do. And we have wild-caught, wild-derived inbred and classical inbred, and you can't see the colors here, but believe me, there's nothing to write home about. These animals essentially have very limited genetic diversity. So is the inbred mouse a good model for a genetically diverse human population? I think you know the answer to this. And so we've been looking very carefully at ways to diversify our mouse models in order to be able to produce better models of disease, better sources of cells for organs on chips, and better ways of looking at drugs. And in doing so, we have selected 8 inbred founder lines, and I should say this started long before I got to the Jackson Laboratory, but I've been dealing with these animals for about 10 years now. And you then breed them back and forth with each other in a very controlled way until you have a rainbow of different alleles that are mixed up between those 8 lines. And this is known as the collaborative cross. And once you've got a really good mix-up, you start doing brother-sister matings to funnel these animals back into inbred strains so that you can get reproducible results between members of that funnel, but each funnel has a different combination of the same set of alleles. And we have about 70 of these strains available. And these have been used in a number of very interesting, studies on human disease, including the one on the left, lower left, which Tom might be interested in which we were able to sort of predict whether we were going to see Ebola pathogenesis or resistance on these animals. Here's just an example of some data. This is a collaborative cross group. We put the same amount of doxorubicin into these animals and looked for cardiotoxicity, which is a major side effect of this cancer drug, and discovered huge differences between them. So in my lab, we've done a kind of a heroic task of giving these animals myocardial infarcts, which is hardly a high throughput screen, if you know what that entails. You have to open up the chest and tie off the descending artery of the animal to get a bona fide infarct. This was done by a postdoc in my lab named Kati Salimova, who deserves a sainthood, and has at this point done about 36 of these lines, 20 experimental, 20 sham, and then looked at all the results. So it's quite a large data set. I'm not going to show you any of the data itself. I'm just going to give you the punchline, and that is that here are 5 of our strains that we looked at, which represent the parental strains. We started with the collaborative cross-parental strains, and we already have huge differences between them in terms of their capacity to resolve scar or survive. And when we looked at the 32 recombinant inbred strains, we saw even bigger differences. In fact, BOMGB on the left there is an incredible mouse. You can do anything to it, and it doesn't die. The scar is well controlled, the ejection fraction is maintained, whereas poor old tofu on the right is a disaster, has huge scars, immediately proceeds to dilation. And so everything in between was there, and we were able to pull out all sorts of wonderful models, including including dilated cardiomyopathy. Here's just a little bit of a soupçon of the data that she's collected. It's rich and it's varied, and you can see that just about every parameter we looked at varies between these animals. And we see that they vary in different ways, so that if you look at survival and you compare that to the amount of scar and dilation, which is severity of your response, or a punch in the ear to see whether it'll heal better, to see how much everything's hanging together in these animals, you can see that nothing adds up. And that's because we believe these are distinct genetic pathways that can be dissected out using this resource. And I'll just give you a couple of examples. So we have a very nice QTL for cardiac rupture. It turns out to be an appendage protein for regulating ciliogenesis. This seemed like a kind of an outlier until we realized that it's already been reported as something that's very important for congenital heart disease, and now we're looking to see what's wrong with that animal. Another one here, chromosome 5, left ventricular dilation, a huge problem in cardiac response to myocardial infarction. And we picked up an unexpected gene which has been implicated in fast skeletal muscle sarcomere assembly, and oddly enough, is expressed in the heart, and it turns out has a very strong role in holding the myofibrillar structure together. So if your heart isn't really very well put together, the first thing that'll happen when you're injured is it'll blow up. And so this actually makes sense, and guess what? There's even a patient described with a mutation in this gene. So we believe that this is a real rich pickings for finding new pathways to understand exactly how this works. And then what will we do with this? Well, the minute you have some candidates, you can go into our knockout resource, you can pull that mouse out, and you can start mating that into this modifier garden, and essentially start pulling out gene interactions. The thing that's so cool about this is how fast it is. And we've made it even faster by producing an even more outrageous resource called the diversity outcross mouse. Now what we did here is we took all the collaborative cross mice, started breeding them, and never stopped. So by the time you're finished with this wild breeding schedule, every mouse has a different genome, and every mouse is an individual. But we know exactly which parts of each part of the genome comes from which mouse. So they're completely sequenced and completely SNP mapped, so we can very rapidly take an individual mouse genotype and figure out exactly which parts of the allelic spectrum it has in it. The SNPs are right up there with human diversity, so it's to the point now where we can start looking at uniqueness. And indeed, I'll give you a few examples. You can't see The mouse on the left is not actually mauve, it is brown. But the body weight is different. You can very rapidly map things like, in this case, chemotherapy, induced neutropenia with DO mice. This is work from Dan Gaddy at the lab, showing 2 different genes that have come up. It's not as if this is necessarily easy to do. This takes hundreds of mice and a very good phenotyping facility, but it's fast. Within 3 months, you can have 4 or 5 different markers to work with, and it's totally reproducible, which is sort of counterintuitive. So if you take 200 mice and you do this study on those 200 mice, and then you take 200 other mice and you do the study, you get the same result. You get the same QTLs because of statistics. And that's what you can do with a controlled set of alleles, which unfortunately we do not have in the human population. So to finish up, what we are looking to do at the Jackson Laboratory with this resource is to functionalize the Human Genome Projects around the world. And the reason this is important is that now we're looking at genetic variation that boggles the mind. And this is something that's now being taken up all over the world. I mean, 1,000 Genomes is here, but we have the 100,000 Genomes England project, and over again, reams of data is coming in. Mutations galore, GWAS data. What are we going to do with it all? Unless we're able to find a way to very rapidly test every one of these complex genetic combinations, we're never going to be able to get the Rosetta Stone of what it all means. And so our feeling is that mouse diversity pipelines are now to the point where we'll be able to actually do gene association mapping and get the variable genotypes and understand them, but more importantly, to understand what it is that mutates when someone is susceptible to a disease to allow us to look at better therapeutics, and I'd say even as a source of cells for Don's organs on a chip. So my own goal in this is to try to figure out what is the difference between someone who dies right on the spot after a heart attack and someone who lives very, very well. And I feel that if we can look at the variation in cardiac repair in a whole organism, which is hard to, with all the immune implications, which is hard to study unless you know something about the genetics underneath and look at the immune composition of that same animal and understand how it's interacting with the interstitium, it's going to be very hard to interpret clinical outcome. So that's my talk. Timing is everything. Early resolution of inflammation seems to be the trick to keeping yourself from getting into trouble with fibrosis. Obviously, the immune response is a huge inflection point, but I believe that we have to look at the way the immune system works with the interstitium to understand better how it is that organs form and function in the first place and how they fall short when they're injured. And obviously, I wouldn't be at the Jackson Labs if I didn't believe in a little bit of genetics. It is a genetic tractable trait, and we're very excited about getting more data on this as we move forward. So those are my lab members. I've tried to mention them as we went along. I have some wonderful collaborators and support. And I know it should be a mouse, but how can you resist that cute little axolotl? Thank you very much.
Hi, beautiful talk. Thank you very much. The point that you raised about the need of genetic variation really brings the question you started with even stronger. So if you use a mouse as a model for a human, but the genetic variation that you get is never the same exactly as what you get in human, how do you actually come to solve this issue? You always have rare issues that are particular to one race or the other.
I think I caught what you were saying, but if I didn't, let me know. What I understood, it was just hard for me to hear. What I understood is you're asking, a human doesn't have the same genetic makeup as a mouse, no matter how diverse the mouse. So what we're finding is that the mouse is nevertheless a mammal. It's wired more or less the same. So whatever you're going to pull out there may not be the same gene variant as is working to create that phenotype in a human. But if you dig into the system's biology of how that gene works, you're going to end up with a network. And you'll pull on it just like Don's tensegrity, and you'll find the gene in human is connected to it.
So what will be your statistical test to verify that indeed you got enough variety from the mice?
Well, I think we've got obviously this is early days because this resource has only just really come online, but what we're finding is that more often than not, at least the obvious QTLs with really high LOD scores map directly onto human. The more complicated part is going to be when we're looking at subtle additive effects, and there is where I think we're going to have to use systems biology to figure out where we are in the molecular biology of the phenomenon.
Well, it's very exciting to see the fibroblast as a target of, a therapeutic target. Can you comment on, and to equate them with a stem cell equivalent reservoir ready to respond to an injury, can you talk a little bit about whether they, in fact, should be viewed that way, as a reservoir that's tissue specific, ready to respond, or do they have a dynamics? Do they come into the tissue in a dynamic way?
So that's a great question. First of all, I didn't mean to say that all fibroblasts will now take over the stem cell literature. What I'm saying is that, at least in the cardiac context, we have no obvious evidence for, at least with the markers, we now have a cell population that is specific to the heart to make new cardiomyocytes. Everything we see are progenitors that make new fibroblasts. So people aren't very excited about studying fibroblastic stem cells, but in fact, it may not be such a boring topic in the long run. Now, other organs, such as the brain, where there are obvious stem cells, we know about those stem cells. My sort of tentative working picture on this is that in terms of the interstitium, I think that I don't think that any cell is completely immobile. That's obviously not the case. We see them wandering around all over the place, and certainly the immune system wanders all over the place in the brain. We saw some beautiful work in Seattle last week with that. However, I think in terms of the stem cells, there's been a kind of facile assumption that once you find a stem cell, it'll know exactly what to do, it'll know exactly what to become, and it'll know when to do it and where to do it. And I think all of that is up for grabs. I think actually it's the environment that's telling those stem cells how to behave. And so unless we know how to put the stem cell in the right environment, you're sticking a man in the Gobi Desert without an iPhone.
Thank you so much.
You're welcome.